terakhir kalinya. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam
Waalaikumsalam warahmatullahi wabarakatuh.
Hello guys, how are you today?
I'm great.
I'm fine. How about you?
Yes, I'm fine. I hope you are always healthy and always happy. Today, we from the group of 13 would like to present the material we are working on, which you are most likely familiar with, namely for simple chemical tests for food. Let us introduce ourselves first. There is me, Adistania Pitaloka, Alivia Anissa, and Bagus Arif Ramadan. Okay guys, please say hello to audience.
Halo.
Hello.
Okay, we will start to explain the material we have discussed regarding for simple chemical tests for food. Simple chemical tests can identify many important compounds in food. Some tests measure the presence of a substance in food, while others can determine the amount of a compound. Examples of important tests are those for the major types of organic compounds, carbohydrates, proteins, fats, and vitamin C. Next. Here are step-by-step instructions to see if foods contain these key nutrients. The first content is Benedict's solution. The second content is biuret solution. The third content is sodium tristate, and the last, the chlorophenol indophenol. Okay, the first content is Benedict's solution. Carbohydrates in food can take the form of sugars, starches, and fiber. Use Benedict's solution to test for simple sugars such as fructose or glucose. Benedict's solution doesn't identify the specific sugar in a sample, but the color produced by the test can indicate whether a small or large quantity of sugar is present. Benedict's solution is translucent blue liquid that contains copper sulfate, sodium citrate, and sodium carbonate. How to test a simple sugar with Benedict's solution? The first, prepare a test sample by mixing a small amount of food with distilled water. The second, in a test tube, add 40 drops of the sample liquid and 10 drops of Benedict's solution. The third, warm the test tube by placing it in a hot water bath or container of hot tap water for 5 minutes. And the last, if sugar is present, the blue color will change to green, yellow, or red, depending on how much sugar is present. Okay, next presentation, I will give it to Bagus.
Okay, thank you, Adis. Okay, for the next, we have biuret solution. Okay, protein is an important organic molecule used to build structures, aid in the immune response, and catalyze biochemical reactions. Biuret reagent may be used to test for proteins in food. Biuret reagent is a blue solution of alphanamide, cupric sulfate, and sodium hydroxide. Okay, how to test a protein with a biuret solution? The first, place 40 drops of liquid sample in a test tube. Next, add 3 drops of biuret reagent to the tube. Swirl the tube to mix the chemicals. Next, if the colors of the solution remains unchanged or blue, then little to no protein is present in the sample. If the color changes to purple or pink, the food contains proteins. The color change can be a bit hard to see. It may help to place a weight index card or sheet of paper behind the test tube and to 8 February. Okay, for next, Sudan tree stain. Fats and fatty acids belong to the group of organic molecules collectively called lipids. The lipids differ from the other major classes of biomolecules in that they are nonpolar. One simple test for lipids is to use Sudan tristains, which binds to fat but not to proteins, carbohydrates, or nucleic acids. You'll need a liquid sample for this test. If the food you are testing is not already a liquid, puree it in a blender to break up the cells. This will expose fat so it can react with the dye. This is how to test fat with Sudan tree stain. The first one is add equal volumes of water, can be tapped or distilled, and your liquid sample to a test tube. Next, add 3 drops of Sudan tree stain. Gently swirl the test tube to mix the stain with the sample. The last one is set the test tube in its rack. If fat is present, if fat is present, an oily red layer will float to the surface of the liquid. If fat is not present, the red color will remain mixed. This is the simple test for fats. Another simple test for fats is to press the sample onto a piece of paper. Let the paper dry. Water and volatile organic compounds will evaporate. If an oily stain remains, the sample contains fat. This test is somewhat subjective because the paper may be stained by substance other than lipids. You can touch the spot and rub the residue between your fingers. Fat should feel slippery or greasy. Okay, for the next, I'll give it to Olivia.
Okay, thanks, Bagus. For dichlorophenolindophenols, chemical tests may also be used to test for specific molecules such as vitamins and minerals. One simple test for vitamin C uses the indicator dichlorophenolindophenols, which is often just called vitamin C reagent because it's much easier to spell and pronounce. Vitamin C reagent is most often sold as a tablet, which must be crushed and dissolved in water just before performing the test. This test requires a liquid sample, like juice. If you're testing a fruit or a solid food, squeeze it to make juice or liquefy the food in a blender. How to test for vitamin C? First step is crush the vitamin C reagent tablet. Follow the instructions that came with the product or dissolve the powder in 30 milliliters, 1 fluid ounce of distilled water. Don't use tap water because it can contain the other compounds that could affect the test results. The solution should be dark blue. The second step is add 50 drops of vitamin C reagent solution to a test tube. And the last step, add a liquid food sample 1 drop at a time until the blue liquid turns clear. Count the number of drops required so you can compare the amount of vitamin C in different samples. If the solution never turns clear, there is very little or no vitamin C present. The, the fewer drops required to change the color of the indicator, the higher the vitamin C content. If you don't have access to vitamin C reagents, another way to find vitamin C concentration is using iodine titration. The summary, some tests measure the presence of a substance in food while others can determine the amount of a compound. Examples of important tests are those for the major types of organic compounds, which is carbohydrates, proteins, and fats. some simple chemical tests are. Test 1 is using Benedict's solution to test simple sugars such as fructose or glucose because carbohydrates in food can be sugars, starch, and fiber. So in other words, Benedict solution is also used to test carbohydrates. Test 2 is use Biorite solution to test protein. Test 3 using Sudan tree stain to test for fat because Sudan tree stain can bind to fat but not to proteins, carbohydrates, or nucleic acids. Test 4 using dichlorophenolindophenols to test vitamin C. Okay, I'll give the time to Adis.
Okay, thanks, Via. So many presentations of the material that we have conveyed. Maybe until here, we apologize if there were wrong words and action. And the last, wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam
Waalaikumsalam warahmatullahi
warahmatullahi
wabarakatuh.
wabarakatuh.